This lesson covers the topic of bacteria. This lesson is to be presented in conjunction with hands-on lab activity. This lesson was prepared by Josh Minich and Sam Drup as part of the National Science Foundation-supported Boat of Knowledge in the Science Classroom. Prokaryotes. Many scientists think that the first organisms on Earth were microscopic unicellular organisms called prokaryotes. These organisms are incredibly diverse and occupy almost every single surface on the planet, even hostile environments such as hot springs. There are two domains of prokaryotes, bacteria and archaea. Bacteria. Bacteria are some of the most studied organisms and are found almost everywhere except for extreme environments where mostly archaea are found. Bacteria have a strong cell wall that contains peptoglycan. Bacteria are found in almost all environments. There is estimated between 500 to 1,000 species in the human gut alone, and there are 10 times the number of bacterial cells than human cells in your body. Archaea. Archaea are generally considered to be extremophiles. They exist in extreme environments that are hostile to most other forms of life. Archaea can be broken down into three main types. The first is thermoacidophiles. These are organisms that can live in both hot and/or acidic environments. Second classification is called halophiles. These organisms can live in very salty environments, such as the Great Salt Lake, which is 5 times saltier than the ocean, or the Salton Sea in California, pictured below. This sea is so salty that only archaea can be found. The third type are methanogens. These are archaea that live in anaerobic conditions and produce methane. These organisms are most often found in the guts of animals and humans. Bacteria and archaea have many differences that have led them to be classified in different domains. Based on their classification, we understand that bacteria and archaea are as different from each other as they are from eukaryotic cells. Some differences include bacterial cell walls contain peptoglycan, lipids in their plasma membranes, and different ribosomal proteins and RNA. Prokaryote structure. Prokaryotes are microscopic unicellular organisms. They have the same characteristics of all cells such as DNA and ribosomes, but they lack a nuclear membrane. Chromosomes. The chromosomes in prokaryotes are arranged differently than the chromosomes found in eukaryotic cells. Their genes are found on a large circular chromosome in an area of the cell called the nucleoid. Many prokaryotes also have at least one smaller piece of DNA called a plasmid, which also has a circular arrangement. Capsule. Some prokaryotes secrete a layer of polysaccharides around the cell wall, forming a capsule. The capsule has several important functions, including preventing the cell from drying out and helping the cell attach to surfaces in its environment. The capsule also helps prevent the bacteria from being engulfed by white blood cells and shelters the cell from the effects of antibiotics. Pili. Structures called pili are found in the outer surface of some bacteria. Pili are submicroscopic hair-like structures that are often made of protein. Pili help bacterial cells attach to surfaces. Pili also can serve as a bridge between cells. Size. Even when using typical light microscopes, prokaryotes are small when magnified 400 times. Prokaryotes are typically only 1 to 10 micrometers long and 0.7 to 1.5 micrometers wide. There are many different types of bacteria, and most are harmless. Prevotella species break down carbohydrates and simple sugars in the gut. Bacteroid species break down animal proteins, amino acids, and saturated fats. E. coli are mostly harmless, but some strains can cause food poisoning, urinary tract infections, and meningitis. These are often found in the lower intestines of warm-bodied organisms. Fecal coliform is a type of E. coli. It's commonly found in feces and used to indicate contamination of a waterway. Although not directly harmful to humans, it indicates the potential transfer of pathogens and is part of the water quality index. How does it get into the water? Typically through non-point source pollution, poor drainage, floods, and aging septic systems. Before refrigeration became commonplace in households, Many cultures used spices to help prevent decomposition through bacterial colonization of food. Various spices such as cinnamon and turmeric have been used to preserve the flavor, color, and palatability of foodstuffs worldwide. We will now conduct a lab experiment looking at the effect of spices on bacterial colonization. Today we will be testing the effects of different spices on bacterial growth. For this activity, you will break into teams of 4, and each group will get 2 Petri dishes and 2 cotton swabs. As a group, decide one item in the classroom you are interested in testing for bacteria. Sample the area with both cotton swabs. Transfer the sample to the agar by lightly drawing a squiggly line on the agar with the end of the cotton swab. Lightly dust one of your Petri dishes with the spice of your choosing. Place each Petri dish in its own individual plastic bag. Allow the Petri dishes to incubate for 24 to 48 hours. At the end of the incubation, record any differences you observe between the treated and untreated samples, as well as any differences you observe between what you sampled and what someone around you have sampled.